Good afternoon. My name is Tom Bruner, and I'm the President and CEO of Glaucoma Research Foundation. Welcome to an exclusive update with our Catalyst for a Cure Vision Restoration team. This webinar is a special thank you to our donors for your support of Glaucoma Research Foundation and our mission to restore vision. I'd also like to thank Aerie Pharmaceuticals for their sponsorship of our webinar series and their continued support of glaucoma patients everywhere. We're fortunate to have all four of our CFC scientists join us today to talk about their incredible research to create a future without glaucoma. Dr. Ana Torre attended the University of Barcelona in Spain, where she received a master's in cell biology and a doctorate in neurobiology. Today, she is an associate professor in the Department of Cell Biology and Human Anatomy at the School of Medicine at the University of California, Davis. Dr. Young Hu received his medical degree from the Beijing Medical University, followed by a PhD in neuroscience at Cornell University. He is currently assistant professor in the Department of Ophthalmology at Stanford University. Dr. Jin Duan attended the Johns Hopkins University School of Medicine, where he received a PhD in neuroscience. Today, he is assistant professor in the Department of Ophthalmology and Physiology at the University of California, San Francisco. Dr. Derek Welsby attended the University of California, Los Angeles, where he received his medical degree and a PhD in molecular biology. Derek is an assistant professor of ophthalmology at the University of California, San Diego Shiley Eye Institute. It is my honor to welcome Ana, who will speak first, followed by Young, Jin, and Derek. After the presentations, we'll have a discussion and respond to some of the questions that were submitted. Ana, please begin.
Thank you so much for the introduction, and thank you and welcome everybody. Thank you for taking the time for being here today. So I'm gonna give you an update of what we've been working on the last years and what are we currently doing in the labs of the Catalyst for a Cure scientists. But before we start, I want to give a brief introduction and a little bit of background so we are all on the same page. This is obviously an eyeball, and the light enters the eye through the pupil at the front of the eye. And as this happens, this light gets focused on this beautiful structure at the back of the eye that's called the retina. If you could see the retina the way we do in the labs with the spatial microscopes, you would see that the retina is made by three different layers of neurons. So there's the photoreceptors, these are the cells that can detect light. And then there's a circuitry of neurons that process this information that's finally captured by these cells here in red called the retinal ganglion cells. So the retinal ganglion cells really form sort of a mosaic that covers all this surface of the eye. And each one of these neurons has a very long, we call it axon, but it's kind of a cable. It's a fiber. And each one of these cells send their axons, these fibers, towards the center of the eye, where all these fibers get together to form the optic nerve. So here we can see a real picture looking inside of an eye. Each one of these dots, green dots, is one of these retinal ganglion cells. And you can see how all these axons are getting together at the center of the eye at this region called the optic nerve. So as light enters the eye, all the visual information is captured by these retinal ganglion cells, and this information travels through the optic nerve, through these cables, and this is the only connection between the retina and the brain. And so when something happens to these cells, something's happening to the optic nerve, that leads to vision loss. And this is exactly what happens in glaucoma. There's several problems that can happen in the eye, most often here in this region, the optic nerve head, this region at the back of the eye. Sometimes this is caused by increased intraocular pressure, but that's not the only cause that can lead to glaucoma. But what causes glaucoma at the end of the day is the degeneration of these cables, this optic nerve, this connection between the eye and the brain. And this happens progressively. It's not sudden most of the times, but this degeneration can cause the death of these retinal ganglion cells. And once these cells are lost, the eye does not have any mechanisms to replace them. And that's really what we perceive as vision loss. So what are we trying to do to really work on this problem? So as in life, time is everything. And so depending on when we act, probably the approach should be different. So if we can act when the cells are just starting to degenerate, the best approach would be to try to preserve them. That's what we call neuroprotection, which is really meaning try to protect the cells from the problems and the insults that may be happening in the eye to prevent them from dying. If we try to intervene, sort of intermediate progression of the disease, meaning in a point where the cells are still there, they're starting to be sick, and these cables are starting to degenerate and to shrink and to fall apart, then at that point, what we try to do is to preserve the cells that are there and to try to get these axons to grow back, these fibers to grow back and reconnect with the brain. Unfortunately, when the cells are lost, that's not going to work. And so the last approach, and that should happen, or that's a strategy that we like to think when there's nothing else to do, is when the cells are gone and there's no retinal ganglion cells left to try to survive. And at that point, really the only strategy left is to try to replace the cells that are lost with healthy cells. So what we are doing together as a team is try not to restrict ourselves to just one of these strategies, but really tries to advance research and science in all these fronts and try to see what can we do to improve neuroprotection, regeneration, and also cell replacement. So this is us. And so in my case, I'm going to tell you about the work we are doing in the most challenging probably part of this, which is trying to replace the cells once they're no longer there. And so I'm saying that this is challenging. And so what are the challenges? We need to replace the cells. So we need a source of healthy cells to inject or to transplant back into the eye. We need these cells that we inject to survive in that host eye. And we need them to grow these axons, grow these fibers, not anywhere, but towards the center of the eye, all the way the optic nerve, and establish the right connections with the brain. So as you can imagine, this is not a simple task, and there's many hurdles that we need to figure out in the way. So the first one is obviously we need a source of donor cells to be able to do these transplants. And so in my lab and many others, we figured out a way to use the stem cells for this purpose. So we have pluripotent stem cells, and pluripotent, what it means is that we have these cultures of cells in the lab that have the potential to become any cell in the body. And we know exactly how to manipulate them and what we need to add to these cultures to make them into retinal cells, including retinal ganglion cells. And so this is how they look in the lab. These are tiny, really little balls, we call them organoids, and they really are very similar to a retina. They have the same cellular composition of a retina. What's interesting is that we can make retinal ganglion cells from these cells that have many properties similar to the normal retinal ganglion cells in the eye, including the fact that they can extend these axons, these fibers, in a dish, and so we can see and monitor how this happens. So what we've been doing the last few years is engineering these cells, so it's easier for us to work with them, so we can monitor many aspects of these cultures. So something we've done is we have created cells that have this red fluorescent protein only in the right type of cells. So in these cultures, only the cells that become retina are going to express this red protein, so we can really observe them in culture. And so this is the right how they look. They are really small. They are not the size of an eye, but these little balls of cells contain retinal ganglion cells. And so why we want them to have this red fluorescent protein? Now we can monitor exactly what parts of these organoids are making retina and what parts are not. And these allow us to have very easy ways to sort the right cells from the cells that are not the ones we want. So now at this point, we have the retinal organoids, and they're very useful for many aspects. We can use them for drug screenings and try to find drugs that improve survival, to model disease. But really what we want to do is to use them as donor cells for replacement therapies. We have those, but there's some challenges still. Only a small portion of the retinal cells that we make are retinal ganglion cells, and we're trying to improve them. But one of the biggest problems is that these cells die over time in vitro. We can make them, they're good for a period of time, and then slowly they degenerate. And so together with Dr. Derek Welsby, and he'll tell you more about this in a minute, we're trying to find the molecules and the signals that help these cells survive longer and be more resilient to damage. So we can obtain donor cells that are better to survive a transplant. And so some of the molecules he's identifying in the lab not only make the cells more resilient, but you'll also see that they also help these cells extend axons. And so that's going to be very useful. So the question is, you can make these cells in a dish. Can we transplant them? And do they survive and work when we inject them in a host eye? So we've done some experiments, and we have a lot of hope because we observe, here the cells are not red, these are green. It's another line that we engineer, but it's very similar. We've seen that after the cells are transplanted in the eye, sometimes they can grow axons, and the axons grow towards the center of the eye. So they are sometimes doing what we would expect, and we've seen that they can survive for quite some time. And I keep saying sometimes because what we detected is that very often these cells die after we transplant them. We can monitor what we do very precisely, and we see that just in a few weeks, for example, these cells in box 1, 2, 3 slowly disappear, and these cells in box 6 and 7 were there in week 1, and they're gone by 7 weeks. And so why the cells die sometimes? We know that mostly is because they cannot cross to the right layer and establish connections with the cells with the endogenous retina that's there. And the reason is because between the vitreous, the inside of the eye, and the layer where they're supposed to make these connections, between that there's a membrane. And so something that we are trying to do to improve the engraftment is to try to mechanically, surgically peel a little bit the membrane to see if this improves the engraftment efficiency. So the tools that we use really looks like a needle, but it has a little forceps, a little mechanical thing at the edge. So we can go into the eye and peel, and I'm going to show you how this looks, a little bit this membrane. We can do it so carefully that we don't affect the blood vessels and we don't affect the cells that are there. And we are now doing these experiments to see when we break this membrane, if this gives more accessibility to the cells that we're trying to transplant. And we only have some experiments, and we're really hoping to ramp up these experiments in the next few years or the next few months to see if this peeling improves these cells that we transplant to survive, and if we can see them growing towards the center of the eye and they can establish the right connections, because only when that happens, we can really have vision restoration. So this is my last slide, and I'm going to let my team members take over. But I really want to thank everybody who's doing the work and the Glaucoma Research Foundation for all their support. None of this would be possible without them. Thank you very much.
Thank you very much, Anna, for the wonderful introduction. And I will jump right into some new tools that we recently developed for study of the neuroprotective and therapeutic strategy for glaucoma. And I want to, first of all, I want to thank Tom and all the members at the Glaucoma Research Foundation that support the CFC3 groups, and also the donors and the patients that trust us to study this very critical, important problem. So to study glaucoma, we want to have something, animal model that is really closely resemble what happened in human patients. So that's why we want to develop clinical relevant glaucoma models. So we got the idea from the patients that got their secondary glaucoma normally can get after retinal surgery, which is using silicone oil to push back the retinal detachment treatment. However, sometimes of the silicone oil can migrate into the anterior chamber to block the pupil to prevent the aqueous humor circulation, therefore to increase the intraocular pressure significantly, cause secondary glaucoma. We mimicked this process in rodent first, and we got a very good intraocular pressure increase, and also characteristic glaucomatous neurodegeneration. And then we extend this animal model into more close to human, which is non-human primate rhesus macaque monkeys. And this right of the figure shows that it really resembles what's happening in human patients. The silicone oil blocks the pupil, and that causes severe IOP elevation and very characteristic change in the monkey eyes in vivo. So here it shows that the optic nerve capping, which is actually for trained ophthalmology, can recognize this very characteristic glaucomatous change on the fundus image of the retina. And but even for untrained eye, we can very easily to appreciate that the optic nerve head is deepening and cupping compared to the contralateral control eye. So this model has really replicated some of the key features of human glaucomas. And more importantly, that we verify the retinal ganglion cell and optic nerve degeneration really happening very relatively fast in this disease model in the non-human primate that we tested. And another thing we developed is that one of the advantage of the visual system is that is the accessibility. So that's why the gene therapy has been initially really focused on retinal disease. And the glaucoma apparently is our target for the gene therapy treatment. To do that, we recently identified a retinal ganglion cell specific promoter called MOS-SNG promoter. This promoter can drive the specific gene expression in retinal ganglion cells specifically. We first again tested in rodent, and then most recently we tested in rhesus macaque monkey, and we can show that the very efficient retinal ganglion cell, especially perimacular region, that has been entirely infected by transgene expression in the RECs, and also the axons that are surrounding that. In addition to those retinal ganglion cells at the perimacular region, and we also detect quite extensive infection and transgene expression driven by this promoter and the AAV2 in the retinal ganglion cell at the peripheral and the mid region of the retina. So that's really give us a boosting of confidence on using this AAV-mediated gene therapy strategy to targeting the disease genes that modulate the gene's behavior to treat glaucoma. And lastly, I want to introduce a technique that we developed is visualize the RGC function. And this is that, using SIO imaging that we can look at the retinal ganglion cell in the live animal fundus image directly. And using this way, we can actually detect that the very early on change or response of the RGCs in response to injury or disease. For example, for the optic nerve injury, in response to those injuries within several minutes, there are dramatic calcium influx in the retinal ganglion cell and their axons, and a long time they decrease slowly after 10 minutes. And this initial response that cannot be appreciated before that without this in vivo imaging. In addition to that, we can really evaluate the RGC function by giving this visual stimuli. In this case, we give the light stimulation. So here is a video I want to show you is that on the left side is the steady state of the retinal ganglion cell in the live animal. And on the right is after give light stimulation, you can appreciate that the different RGCs response by a change of the fluorescence of the calcium sensor. So based on this, we can really study longitudinally in the same animal, same group of animal at different stage of the disease to look at their function and their response to visual stimuli. And we can acquire some unprecedented information through this technique to understand how the RGCs response to the injury and the disease in terms of their function activities. And more importantly, this technique will allow us to look at individual RGCs and follow their progression of degeneration along the time after injury or disease. And we can really appreciate that the individual RGCs, their response, their dynamic response, transformation of their response to light stimulation at a different stage of the disease. And using this technique, we will be able to study, after neuroprotection or even transplantation of the stem cell into the retina, how their functional response will be preserved or how they will change in the disease environment. Lastly, since we developed this imaging technique and also collaborate with Dr. Hao Zhang at Northwestern University, who developed this so-called visual OCT system. This is a high resolution OCT that they can detect nerve fiber on the retina in the live animal, not in the animal. Actually, this has been applied in human patients. So this has very high resolution of the retinal layers and also axon fibers on the surface of the retina. By combining SIO and this visual OCT system, we will have the capacity to look at the morphology and the function of RGCs in response to disease and at a different stage of the disease, and also how they respond to the neuroprotection, regeneration, or stem cell treatment. So I will stop here and thank you very much for your attention.
First, I would like to take this opportunity to acknowledge the Glaucoma Research Foundation, Tom and the advisory board for this wonderful opportunity, and also thank all of you for the generous donation to support this great initiative. As my team member Anna and Yang stated earlier of their presentation, this is an amazing team to work together and to really bring this one goal, that is to restore vision using this team approach. This is indeed a team effort where 4 investigators across 4 universities are working together. So as you've just heard from the introduction from Anna's talk, we took this different approaches to appreciate the cell diversity and also try to provide neuroprotection and to replace the cells. So my contribution to the team is on the relatively upstream in order to understand what are the players there and how we can really provide some innovative and also some bona fide cues and receptors that may allow us to do this in the live animals as a model. So to team up with Dr. Yang Hu's laboratory, we generated a large cohort of experimental animals and also adapted his experimental glaucoma model, which is called micro-beast model here, in order to establish glaucomatous conditions in the experiment models, where we can use one model to examine how the glaucomatous conditions progress across the time and also across different population of the neurons we are studying. So as you can witness, this was first established as the CFC3 started the teamwork together. We had a great fortune to work as a team and go across campuses and learn this technique and adapt it to our experimental models in the mice. So with that approach, basically my team members were able to scan through many genetically marked retina ganglion cell types in vivo using this experimental approach, where we achieved a very large scan of many type of cells of different flavors and at their native states and at different time point of the disease progression. Using this approach, we were able to quantify and also to establish a baseline for my laboratory, also for the team to appreciate that there are differential responses from different type of ganglion cells to the glaucomatous conditions. To take you one example here, where there are a very small type of ganglion cells that greatly reduce their numbers in response to the glaucomatous conditions. We call them susceptible ganglion cells, and there's a gene name associated with it where we study the gene profiling of these cells. But reversely, there are cells that demonstrate this very unique, very elaborated dendritic upper and somatocytes. They are very resistant to the glaucoma treatment. So taking this as a baseline, as a direct collaboration with Yang Hu's laboratory at Stanford, as he just introduced you, that there is this very nice in vivo microscopy that was established by the Glaucoma Research Foundation and the previous CFC's team, where we took advantage of this microscope and really went into the same experimental animal and observed the ganglion cell progression subject to glaucoma over time. As you may appreciate, over a longitudinal observation repeatedly back to the animal eyes, where we can see the changes of the cells over a prolonged time, where in this particular showcase, this population of ganglion cells experience very limited loss subject to glaucomatous conditions, where across the entire visual field, you only see 3 out of 100 cells that go through the loss, even though the glaucomatous models has been established. Using this as a baseline, basically, we have been asking the following question. What are the potential molecular cues, or in other words, proteins that make these cells super resistant to the glaucoma model? And can we utilize these proteins or these cues to teach or to coach the other susceptible cells to prevent the cell loss? So in the experimental setting, we have been had a great fortune to collaborate with Derek Wilsby's lab at UCSD. We learned how to engineer this in a genetic setting to combine the genetic marking and adenosine viruses to generate CRISPR, like a molecular scissor, against the endogenous proteins. And also to work with Yang Hu's laboratory, we were able to generate adenosine viruses to bring this protein back to the original eyes. So then this is essentially serve as a reciprocal approach where we can not only test genetically what's the cause, but also provide potential cures to prevent the cell loss. To give you one example, given the limited time, to give you one example, we bring back the molecule called osteopontin where we studied, and to demonstrate that when we really eliminate the proteins from the endogenous eyes, we can convert the resistant cells to the susceptible form. And reversely, when we really render the cells back with the entire animal with the same protein, and they provide a neuroprotection, even though they came in a susceptible form. So to make a long story short, basically, the past close to 3 years of collaborative work have demonstrated a teamwork where we identified a resistant factor that may explain why certain cells are more susceptible or less susceptible to glaucoma. And our ongoing work is to bring this not only experimental animals, but also use it as a biomarker, as a focus to expand that to the clinical setting. So a very talented resident in the laboratory, Dr. Kinde, has taken this approach where he has been using this as a biomarker initially from the post-mortem human donor samples and show you that these proteins are endogenously localized in a very small subset of the human retina, and also subject to the it's a very clever experiment where we focus the same protein and look into the glaucomatous condition progression over hundreds of patient vitreous samples he collected from the bench side, where we can show indeed there is elevation in terms of extracellular form of osteopontin subject to the progression of the glaucomatous neuropathy. So to summarize, basically our goal here, as supported with the general support with the CFC3 Glaucoma Research Foundation, is to identify risk factors or resistant factors where we can eventually aim at a gene therapy to deliver this and to decrease the glaucoma's damages. With all that, I would like to acknowledge the team members at my junior laboratory, but also the great collaboration with the CFC3 team and also the ophthalmologist on campus at the UCSF. And again, this is a great effort thanks to the generous support and all of you here in the audience. Thank you.
Good afternoon. All right. I'm Derek Wellesby and the fourth member of the Catalyst for the Cure. I want to also be the fourth one to say thank you to the Glaucoma Research Foundation, to the audience, and also to my other three on the team. They're great scientists, and you can tell that from the work they presented, but also great collaborators and friends. I want to talk about the work we've been doing here in San Diego. I know you've heard some of this before, but I'm going to give you another refresher here on glaucoma just because it helps the talk flow if it's fresh in your mind. So what you're seeing here is an actual photo of a retina. This is what your ophthalmologist sees when he or she examines your eye. And I've overlaid a cartoon with 12 of these optic nerve cells and the visual field that the patient sees. In glaucoma, those optic nerve cells get damaged. The cells degenerate and die, and portions of the retina are no longer connected to the brain, leading to vision loss. This continues to spread, and patients lose more and more vision. And sadly, for some, this can be total blindness. So our first goal was to identify drugs and drug targets that could slow or even halt the progression of this disease. To do that, our lab uses high throughput screening using mouse optic nerve cells. We seed the cells into these multi-well dishes, as shown here. And then we use high throughput genetics to turn down the expression of each gene in the genome one by one, going through the entire genome. As in glaucoma, when we injure these cells on the plate, they die, just like in glaucoma. And we're interested in those genes whose inhibition improves their survival, because that means that gene could be a good drug target. So we image the cells and see what we get. And when we've done this, we've gone through 20,000 genes in the mouse genome. We found that the top hits were a pair of genes called DLK and LZK. And this was exciting, but this was also on plastic, and we wanted to make sure this was true in an actual animal. So we turned to a mouse model of glaucoma. In this mouse model, we injure the optic nerve. This in red are the optic nerve cells in the rodent retina before injury. You can see lots of cells. 2 weeks after injury, you see that there's like 90% death. But if we've inhibited DLK and LZK, the two genes we've identified from the screen, we get profound protection. Trent Watkins at Baylor showed that optic nerve cells can be coaxed into regenerating fibers. It's not great, but you can get some regeneration. But interestingly, If we disrupt DLK, if you inhibit DLK, you don't get that regeneration. This is an important point. That means that if we inhibit genes like DLK, we get great survival of injured optic nerve cells, but we prevent regeneration. So why is that? When the optic nerve gets damaged, DLK is actually part of the sensor, and it takes the message back to the nerve cell's command center and tells it to commit suicide. So if the cell doesn't know about DLK because it's been blocked or inhibited, then you can have injury and not have the resulting cell death. But at the same time, if it doesn't know it's been injured, there's no reason to regenerate that nerve fiber. So that's why we also see a lack of regeneration. But if we want to do replacement therapy, like my first three colleagues have talked about, we need to regenerate those fibers so that they connect back to the brain. So Amit Patel, a talented postdoc at the time, now a scientist in my lab, asked whether we could find genes that improve the survival and the regeneration of these optic nerve cells, and if we could do it using human cells. But first we needed a source of human cells, so we turned to stem cells. Blood was obtained from a glaucoma patient, converted to stem cells, and then genetically modified, like Dr. Torre mentioned. We produced these little mini-retinas in a dish, and the optic nerve cells there are shown in red. We can then purify the optic nerve cells and study optic nerve cells from a patient. And this time, we add different drugs to each of these wells. Each of these drugs inhibits a different spectrum of genes. And we just go and add different drugs to different wells. And then we injure the cells, let them die, and we measure things about those cells. For instance, we measure which ones survive. And because we're interested in regeneration, we also measure which ones regenerate their fibers. Now, some drugs help with one and not the other. Some drugs help with both. But honestly, most drugs don't do anything. It's a complicated mess. So what we do is we label which drugs were successful at promoting survival, which ones were successful at promoting regeneration, and then we feed that to a machine learning algorithm. It searches for patterns and tries to identify what makes a drug a winner. And it can do that for regeneration, and it can do that for survival. And it comes up with a list of those genes or proteins that were predictive of having survival activity, those genes or proteins that were predictive of regeneration. And of course, what we were interested in is what's common between those two circles of the Venn diagram. And we found that there was a class of genes called the GCK4 kinases, I'll just call them GCK kinases for this talk, that when you inhibit them, they give you survival and regeneration. But again, this is on plastic, and we wanted to make sure this was true in an animal. So we turned again to that mouse model of glaucoma, where we injure the optic nerve. This time, the optic nerve cells are shown in green, healthy retina there. 2 weeks after injury, you see that profound typical cell death. If we inhibit DLK, we get survival. But I've shown you that before. And if we inhibit the GCK kinases, we also get survival. Now, again, that's great, but we already had DLK, so just getting survival is not the exciting part of the story. We wanted to ask the question, what happens to regeneration? So working with Young Hu, we could coax these cells into regenerating their fibers after injury. So here's a control where optic nerve cells shown in red don't regenerate their fiber. If we knock down a gene called P10, we see a little bit of regeneration. And if we knock down P10 and we inhibit DLK, even that little bit of regeneration is lost. Remember, that was the main problem from the beginning. If you inhibit DLK, yeah, you get great survival, but you inhibit any attempt to regenerate. And then the question, of course, is what happens when you knock down the GCK kinases? When you inhibit them, you don't just inhibit regeneration, or you don't inhibit regeneration, rather you actually increase regeneration. So here we were able to get both survival and increased regeneration. And we were pretty excited about that. So Dr. Torre showed this work before, but just to reiterate, we then took this principle and asked, well, could we then cause those transplanted stem cell derived optic nerve cells to increase their regeneration and to increase their survival. And the take-home message, as she already showed, was yes, we could. So in summary, we've identified genes like DLK and LDK, whose inhibition is robustly protected. And it's probably suitable for patients, or it's being tested, for patients where we just need to slow or halt the disease, but they already have vision, or they still have vision. Chemical genetic screening in these human optic nerve cells has now identified a new class of targets called the GCK4 kinases, where we can get both survival and regeneration. And that might be a principle that we apply to patients where we need regeneration of optic nerve cells. Of course, I want to thank the people that I've collaborated with, Don Zach, Carl Wallen, people in my lab, including Amit. Of course, the Catalyst for the Cure team. This work seriously could not have happened without Young-Hoo, Anna Torre, and Shin Dewan, and the support of Tom, David Calkins, and the rest of the Glaucoma Research Foundation. Thanks so much.
Well, thank you all. That was really fascinating. And we do have quite a few questions that have been submitted, and we'd love for you to answer them. So I'm going to start with the first question, and this is really the tough one. When do you think we might actually see some of these discoveries going to actual trials in real patients?
All four of us knew that was going to be your first question.
Oh, you knew that. Well, that's what patients and donors, and frankly, that's what we all want to know. I think that's the objective of this research, is to be able to help real people who've lost vision. What are your thoughts? And maybe we can just go around and hear from everybody.
So I would give a staged answer. I think you got to think of this problem in two different ways. So there's two challenges, and if we can solve either of these, it's a big deal. But the first is neuroprotection. Can we slow or halt the loss of the cells or the disease course? That is going to happen faster than replenishing lost optic nerve cells. And so that first one, neuroprotection, I mean, there are companies right now that have spun off and are trying to do this. So this is like short term. This is in the next year or two that you're expecting clinical trials. Now, we don't know if they're going to be successful, but I think we're talking about clinical trials in a very short term. Replenishing lost optic nerve cells is a much harder challenge. And there, I could give you a timeline. I'm going to be wrong, whatever number I give you, but I can tell you it's not a year or two, and it's not, 20, 50 years. I'm hoping it's more in the seeing clinical trials in the next decade.
Well, that would be pretty exciting. I mean, if we could really, as you say, I think it's impressive that there could be neuroprotection. And I think there are actually some trials already, limited trials in neuroprotection, and we'll see more of that. So that's extremely encouraging. And then the idea that there could be actual replacement and regeneration, maybe within a decade, within 10 years, that would be really wonderful.
What you might end up seeing is that there might be a bridge to that, where there might be therapies that can increase the capacity of your current optic nerve cells to take some of the load, and that might be an intermediate type therapy that could come out. But again, yeah, we don't know.
I want to add something to this. My lab works with stem cells, and we get asked the question, can you inject cells in our eyes? I sign anything all the time. And as I 100% agree with what Dr. Welsby said, that we won't be there in a year or two. And the reason is because it's not a one problem. It's multitude of problems that we need to solve, and we need to solve them one at a time. And we don't really know what's going to be the strategy that's going to promote survival or the axons to grow correctly. So as we overcome one of the challenges, we need to think about the next one next. But I also want to say that there's some clinics that may offer already stem cell treatments, and these are not safe. Every time you get something injected in the eye, there's risks associated. And all this research with the stem cells for eye diseases are under research right now, and there's none ready for patients yet. So just I want everybody to be careful because you may find a company that offers these for a lot of money, but these have not been approved, that have not been tested, and they're potentially dangerous.
Thank you, Anna. That's a very important point, and I would certainly encourage anyone who might be participating in our webinar today that if you start to investigate something like that, be sure that you talk with your ophthalmologist and your glaucoma specialist, because unfortunately, there are sometimes people offering things that may not be appropriate. So it's very important to be sure that you only look at things that are FDA cleared. And as Anna said, there are none today. There are, interestingly, gene therapies for a different type of blindness. So there's tremendous progress being made. But this is, as you've heard from our 4 CFC scientists, this is a difficult problem. But I also think it's amazing to see the progress that has been made in 3 short years through the technology that each of our 4 scientists are utilizing. So you mentioned, Anna, the challenges, and you talked particularly about we can make new ganglion cells, but how do we actually implant them into the retina in a living animal? How do we get them to engraft into the retina? And you talked about peeling that internal limiting membrane, so these cells could actually send out their axons and dendrites into the retina. What are some of the other challenges that the group faces? I'd be curious to know. I think those are some of the questions being asked, and you've talked about a number of them. But as you say, it's kind of, as you solve one, you uncover another. And I'm just curious what you see as the next big challenge that you're facing.
I'm going to let somebody else talk too, but
well,
I think, yes, I don't know if Shane wants to chip in, but I think learning what combinations of cells is probably something important.
The fact that how many different I think you all have identified, it's a significant number. How many different types of retinal ganglion cells are there?
Yeah, so I thank you, Anna and Tom, for bringing this question to us. So it's actually that's exactly what I want to take over the conversation. So I think one of the biggest challenges is how to translate the knowledge that we learn from all these experimental animals, especially majority of the knowledge, as you saw from today's talk, was gathered in experimental mice, and how to even make the baby step, as Dr. Yang Hu pointed to us, to the monkey model, which is one of the most successful model where, University of California, Davis has one of the world best primate center for a lot of the experiment we launched here. So how to even make that baby step and how to translate the 30, 40 type of mouse cells to a little bit less than 20 type of monkey cells, which is the most closest to human. So one of the last slide I showed using our molecule, right, this CFC3 molecule, osteopontin, where we made that first link. And essentially, that is just a marker. That is just a biomarker. And what it really translates from mouse to human is a functionally conserved thing. So the limited electrophysiology, and most recently a collaboration with Dr. Yang Hu's lab on a functional setting, like calcium-based functional setting, is to tell you those are evolutionarily conserved cells that may respond to a particular type of visual stimuli. And likely our current guess is it's a population of cells that are responsible for the light adaptation. When you walk from an extreme bright room like this to an exclusive dark room, and half of those population cells will fire. And reversely, when you come out of the dark room to a very bright room, and those are the cells that fired most robustly. So it looks like our very basic work, so-called neuroprotective or resistant cells, happen to be this population of cells that exist in mouse and also now in monkey. And as I briefly showed you in the postmortem tissue, we started to detect 3 to 5% of population. So sounds like we are putting a manual together that we are having a very optimistic guess that when this menu reaches to the number of 5 to 8, I think our recipe is there, our cookbook is there. And these are the cells that our team member, Anna, will focus on regenerating and producing. And our drug screen with Dr. Gillespie's work was just focusing on generating chemicals and potential cues to further boost their regeneration and survival. I feel like we do have a solid plan to make that baby step from mouse to human, mouse to primates, and eventually to human. And I will stop there and let our team member to take over. Thank you.
The only thing I would add is, as a follow-up to what Dr. Dwan said, when all of us present about what an optic nerve cell is, we always show a little picture of the cell in the retina, and it's got its long fiber. It doesn't just communicate that there's a pixel of light at that point on the retina. There's a whole series of connections. It's like a little computer that has to process a lot of visual information. So, Tom, to answer your question, I think one of the big challenges is not just getting engraftment, but it's getting all the appropriate connections so it does the correct visual processing to then send that message to the brain, which is yet another challenge.
Well, I agree. So there are many challenges, but I think, to me, one of the most interesting things is the discoveries you're making along the way. And I think you mentioned, Derek, that the potential for maybe preserving or enhancing ganglion cells that are already there could be another whole approach. And if Jin's work shows that there's a particular type of ganglion cell that is more resistant or that can be helped to continue to function, there may be a way to maintain some partial vision for people that have lost significant amounts. And this raises another question, really. The work you're doing, does this apply to all different levels of vision loss, or only people that have totally lost all their vision? How do you see this research applying to the different stages of glaucoma, from the earliest loss to, say, almost complete blindness? Could you speculate on that?
I think the way most people think about the clinical trial design is that initially this is going to be more relevant to people with more severe disease, right? If you're testing something that's new, you want to test it in somewhere where the risk benefit makes sense, someone with very severe disease. But the potential is certainly, we could extrapolate to very early disease. I think this, the neuroprotection is something that we would envision could be something that we apply to anyone with a risk for glaucoma.
I want to just chip in about this. I totally agree with Derek and also Tom, you mentioned. And I think the neuroprotection for the still survival RGCs and the still connected axons, there it may have even more beneficial than what we thought. The reason I'm saying that is that just recently we identified something very strange, that some genes that can promote regeneration, but in the crash model that we don't see it has significant cell protection, which means still dramatic cell death happening, but there's for the survival RGCs can regenerate their axons. However, when we test in glaucoma model, that it has significant RGC protection, cell protection as well. What I'm saying is that, in terms of the first question, when we can do the clinical trial, I'm very optimistic. I think within just 2 or 3 years, we should have very promising neuroprotection target that will be addressed in clinical trial, like something that Derek just mentioned, and also something else is in the field that shows very promising neuroprotection. And those, of course, final proof will comes from clinical trial. But I'm really have high hope with that. And in addition to that, just for the question that Tom just mentioned, even at very late stage, we are not really sure that those cells already totally gone, totally died. So for some treatment, neuroprotection treatment, or even some regeneration treatment, clinical trial, if there's some effect, from early stage patient, it may surprise us that even for very late stage patient, we also can see some beneficial effect, which is there will be some mechanism that can explain that. But for now, before we testing it, we cannot just saying at very late stage, we have no way other than just totally replace the whole thing. I think we may just protect, even for the current strategy, that we protect something that can really beneficial for even at very late stage patient.
Well, I think that you make an extremely important point, Young, and that is that this research, until we get further along, we don't know how broadly it could benefit different stages of glaucoma. And it's all the more reason why this critical research is important, because we're at, every day, every week, we're learning and discovering new things and figuring out how we might be able to protect these ganglion cells, or how we might rejuvenate, maybe restore cells that are still present, as you say, but just aren't functioning. Well, I just want to say thank you to Anna, Derek, Jin, and Young, first of all, for the incredible work you're doing, and secondly, for taking the time to be with us today, and really for your dedication to glaucoma research. I want to also thank all of our participants for their interest and support. And if we weren't able to answer your question today, we will certainly try to answer those questions in our upcoming webinars. The latest information about glaucoma and our research can always be found on our website, www.glaucoma.org, and on many of our social media platforms. Glaucoma Research Foundation has advanced innovative research for more than 40 years. We can do this thanks to our generous donors and corporate partners and their ongoing investment in our work. Today, we'd like to give a very special thank you to our dear friends, Stephen and Michelle Kirsch, for their unwavering partnership and remarkable investment toward a future free from glaucoma for more than 20 years. They have offered to match every gift made between now and December 31st up to $1.5 million. Their generous gift will provide funding for the next 3-year phase of our Catalyst for a Cure on vision restoration. So please join us today and double your impact for Glaucoma Research Foundation. Every gift, even if it's small, makes a difference, and we would be honored to have your support. Thank you also to Aerie Pharmaceuticals for sponsoring this webinar, and thank you all for your attention today and for your interest in Glaucoma Research Foundation. Because of you, the cure is truly in sight.